Okay, I'm gonna go ahead and get started. So if anyone gets on and has any questions, please feel free to just ask me during the lecture and I can stop and answer those. So today, let me go back one slide. So this is metabolism and temperature balance. So really all we're talking about today is how the body is going to use what we absorb from the digestive system, and then how our body is going to maintain its like perfect heat at like 98.6 or whatever that is, right? So the study objectives, very simple today, honestly. So we just need to understand like what's involved with homeostasis of heat, and you guys should already know that. We talked about that like a long time ago. So it's all about the hypothalamus and the regulatory signals that the hypothalamus will send out. So we'll get a little bit into that, but honestly, we won't focus too much on it. Know the basic mechanisms by which metabolism occurs. So like this is a great introduction today. Um, for glucose metabolism, like glycolysis and all that nonsense. But honestly, you guys, if you haven't taken biochem yet, you'll get so in depth. Like I feel like a full midterm is just glycolysis and glucose handling and transport in the body. So we're just gonna like give you the very basics in this class. But in the future, you're going to have to know a lot more if you don't already know it. And if you have taken biochem, then everything here should be just a really easy refresher, nothing hard. You don't need to know all the enzymes or anything like that, but just how glucose is handled in the body. Okay, understand the mechanism by by which insulin is secreted. One, so like what signals for insulin release in the body, and then two, how like what action insulin actually has, because we know like insulin, blood glucose, but how does it do it? We need to figure that out. And then last thing, just know this is pretty simple. Like, so I should have said simple, not easy. Like these concepts can be a little bit hard to grasp, but there's not too much to this lecture. Okay, so we're just gonna go right in on. So heat balance, I'm sure that there'll be a question on this for the digital dialogue or something like that. I'm not going to cover this because it's really simple, right? Like you, any heat that comes in needs to go out, or if we are like an imbalance, right? So it's really cold outside, then we need to like maintain heat in our body. There's a couple different ways we can do that, which will be covered in the next slide. But this is just like an overview that you can like see. Okay, so well, I'm getting heat from the sun. I could get it like conduction, like maybe someone's touching me. I'm touching something hot, like an oven or anything like that. And you also have internal body heat, right? Like metabolism creates heat, and just functioning creates heat. So you need to make sure that you're maintaining that, or else you get a fever, right? Or you get super cold and you go hypothermia. Both of those are super bad. So heat balance occurs in the body. Honestly, that's all you need from here. Okay. Oop, 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 one sec. Okay, so this is like the chart that you would need to know for thermoregulation. And I wouldn't even know it like all that well. I would just recognize the big things in here. So you can have a change in environmental temperature, right? That's like, that'd be like the sun, that'd be like touching something hot, going into a refrigerator, into like a cold, like ice room, you know, like where they keep stuff in restaurants. Like that'd be an environmental temperature change. Then changing core body temperature, that could be something more along the lines of something inside is too hot or too cold, and that's a big problem too. So your body can deal with both of those things. It just does it in a different way. So you either have peripheral thermoreceptors or central thermoreceptors. Those should be pretty easy to like have memorized, right? Central coming from like the center of your body, then it gets all pumped into the hypothalamus. So the hypothalamus will then send out all these different signals. And you can think of it like, okay, so if I am like I have an increase in temperature, what's going to happen? And like these green boxes are going to show you like what the actual action is. These just tell you like what anatomical part of the body actually will do that. So you say I'm too hot. Well, I'm going to vasodilate because that makes it so more heat can get lost, and I'm going to sweat. Like that's super simple, right? Just know that that's like not by happenstance. Your body is telling you to sweat. Like sympathetic system is activated, and I think that's really cool, right? So we just think of this as just being so easy, but honestly, our body's pretty involved in regulating all of this. So I would know from here, like the hypothalamus, thermoregulatory center, sympathetic cholinergic neurons. You guys don't need to know that because that's a little advanced, like, because we learned earlier that really sympathetic system only works through adrenergic, and this is kind of an exception. So I wouldn't know this too in depth. Maybe there'll be a digital dialogue question, but here's what I would know, like this green line. So sweat secretion, vasodilation, vasoconstriction, shivering thermogenesis and shivering thermogenesis. Those both just mean like heat creation. Hopefully you can see that. Genesis, like making something new, and then thermo, heat, okay? And then that's what you get, like metabolic heat, blah, blah, blah. So that is pretty much all the thermal I'm gonna go into for this lecture. Hopefully that's clear enough for you guys, but there shouldn't be anything here that's like too difficult to understand. So energy balance, this one is, I don't know, there's this slide and then this slide I don't completely agree with, but we'll go over it, right? So energy balance, energy in needs to be energy out. If you take in more energy than you output, then you're probably going to gain weight somehow, probably in like white adipose tissue, or maybe in muscle. Like you can gain mass in many different ways, but in most of the time, whatever we take in, we usually will push out through heat or work, like exercising or just basic metabolic rate, which actually burns the majority of your calories. Even if you work out for like 8 hours a day, well, maybe if you work out 8 hours a day, then you're actually burning more calories by active moving than you would be just as your basal metabolic rate. But most of the time, just sitting around doing nothing will burn more calories in general than like exercising, just because your body has to keep um, has to keep you going at all points in times, if that makes sense. Okay, so gonna move on from energy balance. I think it's pretty simple. Then this is the one I don't really love. So um, I'm getting more and more into like the research in caloric intake and how just not all calories are made equal, right? So we think like, well, if 1 gram of fat is equal to 9 calories. I said that wrong. Something, I forgot what that conversion is. And then like 1 gram of carbs is 4 calories and 1 gram of protein is 4 calories. Well, those calories actually are going to have very different effects on the body, but you don't need to know that. That's like my personal rant. So we'll just go through this. So you should know a couple things from here, right? So once again, hypothalamus, and that's kind of why these lectures are put together, because the hypothalamus ties together like feeding, and it also ties together like thermoregulation. So it's easy to clump these two things together, right? Whenever writing books or anything like that. So you just have a bunch of neuropeptides. All honestly, you guys, all this is saying is that the hypothalamus will regulate how much food you intake, how much fat you store. Leptin secretion is a specific hormone that will act on negative inhibition of neuropeptide Y. So you can think of leptin as like you are being satiated if you have a lot of leptin because it shuts down this pathway. If you have a lot of neuropeptide Y, then you'd actually be a little bit hungrier. Um, there's nothing specific that you guys really need to know here besides just generally know if you see neuropeptide Y, if you see leptin somewhere, you're thinking this pathway that goes through the hypothalamus and centers around food intake and fat stores. Okay, Dr. B loves all of his like equations and all his numbers. So you guys can take a look at this if you want to, but honestly, this isn't anything that's important for this course. It might be important for further courses. I've never had to use any of this information before, so I'm not very familiar with it. I'll just be honest with you there. So if you guys want to learn more about this stuff, you can ask Dr. B questions about it, but I'm just gonna go over it. Okay, so metabolism. This is like my bread and butter. I love this stuff. Okay, so this is such a confusing picture. I just wanted to use it because we want to keep the slides the same between what Dr. B is using in class in general and what I'm using. So in your diet, you can take in 3 different macronutrients that will provide calories for you: fats, carbs, proteins, right? And so all this is saying is these 3 systems are actually involved with one another. It's not like you just take in carbs and that's only going to be glucose forever. It can actually sadly be stored and turn into like fat. And then so a lot of really high carb, high fat diets is how we're becoming obese in America. If you can combat that with more protein intake, that's too much. Sorry. But yeah, if you guys have questions about this, send me an email. I'd love to talk more about it with you. Okay. Okay, and a fed state versus a fasting state. So that's just what it sounds like. So a fed state is like, think of you just recently took in food and now there's a lot of nutrients in your blood, right? So there's maybe some fat flowing around in the lymphatic system that's going to be dumped off in the vein soon. There's glucose in the bloodstream and your body can sense all that. You're in a fed state. So when you're in a fed state, you're going to be in anabolism, think anabolic steroids, you're going to be building. And it's not just good building, right? If you are always fed all the time, 24/7, then you're actually going to build a lot of like fat storage. You're going to build probably visceral fat, which can be very dangerous for like heart health. And the visceral fat is just right around your gut. And then the exact opposite, right? So fasting state, catabolic, you're breaking stuff down. And some people are really scared saying like, oh, well, if I fast, like I'm going to be breaking down my muscles. And that's actually been proven not to be very true at all. Your body doesn't preferentially break down muscle tissue. It can actually do many more things before it does that. But that could happen, but it doesn't. So that's kind of cool. Energy utilization. This slide's kind of cool. So this is really the intro into like glycolysis, gluconeogenesis, glycogenolysis that you need to know for this class. So just let's go easy. You have glucose. Glucose can come in. So picture this arrow right here is like coming in through a cell membrane or something. It gets converted to glucose 6-phosphate. Even that fact you guys don't really need to know. And then you go through glycolysis, form pyruvate. Pyruvate gets converted into acetyl-CoA, and it goes through the citric acid cycle. So this blue line, mitochondria, right? That is something I would know. No, glycolysis occurs in the cytoplasm, and then everything else occurs in the mitochondria, okay? Once the pyruvate makes it across the membrane. So this slide, this picture alone would take like 2 to 3 weeks in biochem because you have to know all the enzymes, every single like cofactor, all of that nonsense you need to know. So just know from this class, we don't really care. We just want you guys to know what the general terms are so you can sound smart when you talk about it. So glycolysis is breaking down a glucose molecule, and that can come either from like the blood or glycogen stores. And glycogen is stored either in the liver or the muscle cells, and it can be signaled to be broken down. That's called glycogenolysis, right? Like lysine of the glycogen. And then that gives you more glucose in your bloodstream. So that's glycolysis. Citric acid cycle is just converting acetyl-CoA into like high energy molecules that can be used in the electron transport chain. And then one big question that I want you guys to see right here, which arrow points to gluconeogenesis? Okay, take a second to think about that. What arrow? I only think there's one. If you guys can find another one, let me know. But I think there's only one. So which arrow points to gluconeogenesis? Take a second to think about that, and then I'll let you know. Okay, so this arrow right here is the primary one I think of for gluconeogenesis, but really anything that gets you back to pyruvate. So gluconeogenesis is the creation of glucose, and it only occurs in the liver. So if you're really low on glucose in the blood, the liver will take pyruvate and all these other small like amino acids, because pyruvate can be easily converted into alanine, which is an amino acid, right? So it'll take alanine, pyruvate, and all these other amino acids and push them through this process to make more glucose and then ship the glucose out to the rest of the body, okay? So gluconeogenesis, making glucose, new glucose from things in the body, okay? So that's just what I would know. All these other ones are kind of cool, and Dr. B will probably talk about them in our refresher on Wednesday, but for now, don't worry about it, okay? Fat and cholesterol, this is kind of cool. So this is just a follow-up of the previous lecture, so it's not really anything crazy new, right? So we know what a chylomicron is at this point. So it's like a lipoprotein coat, and on the inside you have triglycerides, you have cholesterol, and depending on like the lipoprotein concentration on the surface, it can be HDL, VLDL, LDL, all that nonsense. Once again, that's a little bit too much for what you guys need to know, but it's so cool. So I really hope you guys get into this someday. But chylomicrons, the proteins on their surface will match with receptors on cells. So then the chylomicron will be, once it matches to a cell, it will be endocytosed. So now that cell is going to start using the cholesterol and the the triglycerides for energy storage usually. So what happens, chylomicron comes in and then it gets broken down by, I think this is lipoprotein lipase. Yeah, lipoprotein lipase cleaves the triglycerides, and then it can either use them for like beta oxidation. So we learned the term glycolysis. Glycolysis is for like glucose breakdown. Beta oxidation, that's just very similar to glycolysis. Just beta oxidation is how we do that with fats, like fat molecules, okay? So how we take the fat apart to actually get energy from it. Once again, that's super taught in biochem, not really taught in this class. But yeah, just know that the chylomicrons come in, then they can like leave. I don't really know what else to say about this. Hopefully this chart is like pretty self-explanatory to you guys. If it's not, let me know. But yeah, chylomicron from the blood dumps off its fat, its cholesterol, then that can either be put into like liver cells down here. Normal cells, we use oxidation, or adipose is like where we store it, right? This would be mostly in the triglyceride form. And then, yeah, I just think that's super cool. Like fat is very highly regulated in the body. Okay, so I'm gonna move on. Metabolism in a fasting state. So the big terms you need to know. So think fasting, low blood glucose levels. That's like, those two things are pretty synonymous. And also when you're fasting, you're gonna have higher levels of glucagon to insulin. We haven't talked about those two hormones yet, but we're gonna talk about them. I just want to introduce them a little bit. So glucagon is the hormone that tells your body to like, tells the liver and the cells to give off glucose because the blood glucose is too low. Um, and just remember that like you need to maintain a very steady blood glucose. Like if you go below like 40 milligrams per deciliter, you can start having like seizures and horrible side effects. And if you get above a certain level, like if you're a diabetic possibly, then you can also have a lot of side effects. So your body is trying to regulate the blood glucose levels, sometimes at the expense of like some of the cells, because some cells can die if the whole organism stays alive, but usually they can just ask the liver, and by they, sorry, I mean your body can ask the liver to rate its glycogen stores, to do gluconeogenesis, and most of the time that's gonna be decent enough to keep your body going, okay? So in the fasting state, that's exactly what's gonna happen, right? So you have glycogenolysis, meaning that like that storage form of glucose is gonna be broken down, glucose is gonna be shipped out, but you also have gluconeogenesis, which are not the same thing, right? Those are two different processes that you need to know are being signaled by like glucagon in a fasting state. So gluconeogenesis, making new glucose, glycogenolysis is breaking down glycogen to like, they were still in glucose molecule form, but now you are like letting those go. And that's pretty much it for what I know in the fasting state, okay? So just remember, you guys can always ask me questions if you have any concerns here. Insulin release is stimulated by what one of these? Think about it for a second, and then I'll give you guys the answer. Okay, so we're gonna go over this, right? So this is just like a preview question. If you don't know it, that's totally fine. Don't feel weird about it, okay? But it's all of the above actually. So vagal stimulation, when you see vagus nerve, you think parasympathetic response. Always, 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 okay? So if the vagus nerve is innervating something, you say, well, if that nerve is stimulated, I'm gonna see a parasympathetic response. If I release insulin, is that parasympathetic or sympathetic? It's actually going to be more parasympathetic because you're pushing, trying to get rid, like it's still a part of digestion, like getting the glucose where it needs to go. Because without insulin, it's just going to stay in the blood, right? So you need insulin to put it off somewhere. Increased blood glucose, that directly goes to the cells. We'll talk about that in just a second, and also GIP from the intestine. Okay, this is probably one of the most important slides or topics for this lecture. So glucagon and insulin. So secreted by alpha and beta cells respectively. So glucagon, alpha cells, insulin, beta cells. And those are just two different cell types in what's called the islet of Langerhans. And if you guys have taken, what is it, like the histology class, then you can literally see these guys in the microscope, right? You see these like big, like they kind of do the circle around here, but you could actually like see specific cells. And then if we like stain it in a cartoon form, you can see like there's beta cells in here. Let's see. I think that the alpha cells are usually around the exterior and the beta cells are a little bit more interior. So both insulin and glucagon are coming from the same place, which I think is pretty cool. So like that same organ is in charge of releasing both of them. So for glucagon, we don't need to know as much besides like, well, if I have really low blood glucose, glucagon's gonna go do its thing. It's gonna do anything it can to increase blood glucose levels. So if you guys see a question on that sometime and it's like, glucagon does which of the following, you wanna say like, which one of these mechanisms will increase my blood glucose levels? Like, does glycogen synthesis do that? Like, no, that actually lowers it, right? So that'd be something that insulin's in charge of. So I'd be able to differentiate between what glucagon's gonna be doing and what insulin's gonna be doing. So insulin is secreted by glucose ATP dependent potassium channel closure. It's kind of cool. So I don't think there's a really good slide There isn't. So I'll just do that really quick. So um, there's this channel that allows like potassium to go out from the beta cells, and it can be blocked by ATP. And when you get, and you have extra ATP, if there's like a lot of glucose, right? So the beta cells actually have a specific kind of receptor that just allows glucose to come into them without like needing insulin secretion, right? Because then you can never get insulin secreted if the cells that produce insulin can't be signaled for it. So if glucose comes in and makes ATP, that channel closes, and that allows like insulin to be exocytosed from the beta cells by like doing calcium intake and all that stuff. And once again, little too in depth for this class. Amino acids in the blood, incretins, and incretins are GIP and GLP-1. We talked about those already. But if you see GIP or GLP, you think that's going to act like insulin, even though I'm pretty sure GLP stands for glucagon-like peptide or something. It acts like insulin, so that's why we always do it in the abbreviated form so it doesn't confuse you guys. But if you ever see that, you know that that is GLP, like glucagon, like protein or peptide or whatever that is. Okay, then the last couple, CCK, gastrin vagus, amplify, and sympathetic inhibition. Hopefully that makes sense, right? If you're increasing the parasympathetic response, decreasing the sympathetic response would probably be occurring at the same time, and they'd have the same effect, right? An increase of parasympathetic activity is equal to or very similar to a decrease in sympathetic activity. So I would know these things for like what lets insulin, what secretes insulin, what tells the beta cells to secrete their insulin. The push-pull hormone function. So we already talked about this. Don't get confused by these graphs. I feel like whenever I see graphs, I get a little bit confused. All we're saying here is that insulin and glucagon work very closely together to regulate metabolism in the body. So like that's actually a level that they're trying to use more and more nowadays, like your insulin to glucagon ratio for like metabolic tests to see how healthy patients are, which is cool. So it's not like really a part of the standard of care yet, but they're exploring that. So all that's happening here is like if insulin goes in a fed state, insulin's going to be dominating. So this is showing like there'll be more insulin. It's heavier in these terms is what this is trying to show. You'll get glucose oxidation. So that's like the breaking down of glucose, glycogen synthesis, right? To get glucose out of the blood, increase in fat synthesis because glucose can be converted to fat, and increase in protein synthesis because like pyruvate, which comes from glucose, can easily be turned into amino acids. And the exact opposite for glucagon. That increases the breakdown of glycogen, that increases gluconeogenesis, and it also increases ketogenesis, which is kind of like breaking down fats, but it's not the exact same. And I think you talk more about that in biochem, but you don't have to talk about it here. So just know glucagon and insulin work together. They're regulated together. I think the term is like they're coordinated hormones, just meaning that one has an opposite action and they respond to similar but opposite stimuli. Okay, so the last portion of this lecture, I think this is at least the last portion, is just understanding the insulin, what insulin is going to do to your body. So first of all, insulin receptor is a tyrosine kinase, and hopefully you remember tyrosine kinases like have two monomers in the cell membrane, and then when they bind their ligand, which is insulin, right? So they bind their ligand, they come together, and then they can send down this phosphorylation cascade. Whenever you see phosphorylation cascade, you think, well, that's going to eventually do second messenger pathway, and second messengers usually will increase transcription factors or change like enzymes, how they work. But what ends up happening here, what we need to know is actually in the next slide. So hopefully this, like, I think all we really need to know here is this is an RTK, or receptor tyrosine kinase, and insulin activates it. And then what you really need to know is here. So this is in most cells in the body, so like muscle and fat cells. What happens is these GLUT4, and I would like know this number because this is how we differentiate. They're all called glutes, or glut. I call them GLUT. We'll see what Dr. B says. But the GLUT4 channel that would allow glucose into the cell is actually just in a vesicle, like waiting to be put on the membrane. And when insulin comes, that cell receives a signal through what we talked about in the last slide, like transcription increase or enzymatic changes. And it gets that signal, and then it comes up and fuses, right? So this little vesicle guy fuses up here with the membrane, and now glucose can be put into the cell. So it's super cool, right? So I don't know why, but that's just so interesting to me that your body just has these channels kind of docked and ready to go. And then if insulin tells them, it'll put them put the glucose channels into the membrane, and all of a sudden you can get a lot of glucose into your cell. So that is the GLUT4 transporter. This is how it functions in muscle and fat cells, but it is very different in liver cells. So you can read that question if you want, but it's called the GLUT2 channel. And what happens in liver cells is the GLUT2 channel is always in the membrane. So GLUT4s are in little vesicles, not in the membrane. They can't let any glucose in. GLUT2s can. So how does this liver actually take care of this? It's kind of cool. So once again, this is a biochem thing you'll learn about. We'll just do the very basics for this class. But what happens is you get insulin, right? So that this is telling the liver like, okay, glucose is really high. You need to start taking care of this, right? Because the liver is like the main organ we need to think about when we think of glucose regulation in the body. So what's happening here is that the hexokinase mediated conversion of glucose to glucose 6-phosphate keeps levels low inside the body. Okay, so once you receive that signal, the liver can actually be told to start making more glycogen and keeping the glucose inside, right? Because if this is just a channel that's always there, depending on the concentrations, it can just flow in or out. So if it changes it to glucose, if the liver changes glucose to glucose 6-phosphate, now it doesn't fit in this channel, and it doesn't really play a part in the gradient here, like high glucose on the outside, low glucose on the inside. It's a new molecule, right? And then in a fasted state, liver cells make glucose and transport out of the blood. So the exact opposite. If you don't have insulin signaling, right, then you're probably having glucagon signaling, which we're not showing here, but you're going to raid your glycogen stores and do gluconeogenesis, so more glucose gets pumped out of the blood, out of the liver into the blood. So sorry. Okay, so I would know that. So GLUT2 always in the membrane, and insulin and hepatocytes, right, in the liver cells actually will like tell you to make more glucose 6-phosphate instead of regulating it at the membrane level. Okay, so that's really cool, I think. And there's obviously way more GLUT channels we could talk about. We didn't talk about how the brain does it or anything like that. That's all you really need to know for now. And if you want to look more into it, you totally can later. Okay, I think this is the last. Oh, I was way off. There's 4 more slides. Okay, glucose homeostasis. Oh, actually, I think we could skip this all because we've already talked about most of this, you guys. So yeah, insulin, good. Yeah, we've talked about all this. So I'm gonna skip this slide. Glucose tolerance test is something that we should know in general. Once again, these are numbers, and you know Dr. B likes his numbers, so don't be surprised if this is one of the digital dialogue questions on Wednesday. But what you're saying here is if you give a patient at time 0, oh, I guess this is the fasting test. There's multiple glucose tests you can do. One, you give them a certain level of glucose and see how it stays in their blood. But I guess this is the fasting plasma glucose. So you can tell with a diabetic patient that they always sit super, super high. So, oh, here it is. Okay, so here's the time after glucose. So right here, you were fasting and they gave you glucose, like a specific amount. And then right here is like 1 hour post. So you can see in a healthy patient, yeah, sure, you get a little bit of a spike, but then you go back down and you're back to like a healthy level, which is between like, I think the lowest you can go is 45 milligrams per deciliter to about 100, but this is showing you right around 80, which is a little high, but still like reasonable. And diabetic patients, they might even sit higher in general, but then even after a long period of time, they still have really high glucose because their insulin is not working. And there's so many reasons like why that can occur. There's a lot of different theories out there right now. Like some think it's because of fatty deposits in the liver. Some think it's insulin resistance on the membranes. And you know what? I'm not sure what the right way is. I kind of lean more to the fatty liver disease, but that's not something you guys need to know. Just know what like A1C is glycosylated hemoglobin. So remember that you have hemoglobin molecules in your blood. You can actually add glucose. Like if your glucose is really high, you'll add glucose to the hemoglobin and they can measure how much of that, like what percentage of your hemoglobin does have glucose on it. So these are all really just telling you methods of testing for diabetes, okay? Type 1 diabetes, no insulin. So this is a crazy, crazy slide. And once again, there'll be a question on this, but we're not really gonna go over it because it's a little too in depth for what we need to know. Like what we need to know is if a type 1 diabetic can't produce any insulin, well, then that means that the glucose is gonna probably stay in the bloodstream. But what actually can happen is the liver can take in all the glucose and not be told to push it back out. So you can, yeah, there's just a lot of issues that are going on with type 1 diabetes. So that's why we give them insulin. We give them other drugs that can stimulate um, insulin production as well. So that's kind of cool. Well, not insulin production. That's a straight up lie. We give them insulin. I was talking about type 2 diabetics. Okay, so fasting state here. We already talked about this. So if you guys have any more questions, please let me know. But I think that's it for now. So I'm gonna like stop this